Hello, my name is Lamry Reynolds, and I will be your instructor today. For today's lesson, I will be talking about anatomy and physiology, specifically the contraction cycle. The learning objectives that I have for you today. By the end of this lesson, I hope that you will be able to draw and label a skeletal muscle at all anatomical levels, from the whole muscle to the molecular components of the sarcomere. I hope that you'll be able to draw the structure of the neuromuscular junction, list the steps in excitation, contraction, coupling in skeletal muscle, as well as describe the roles of the sarcolemma, transverse tubules, sarcoplasmic reticulum, thin filaments, and calcium ions. So let's begin by distinguishing between the three different types of muscles. You have skeletal muscle, cardiac muscle, and smooth muscle. For the sake of this lesson, we'll just be talking about the skeletal muscle. But just as a refresher, remember that the skeletal muscle has large fibers that are multinucleated and they are striated. Cardiac muscle is also striated, even though they are unnucleated. And the smooth muscle fibers, they do not have striations and they do have nuclei. So, yes. So, like I said, specifically, we'll be talking about the skeletal muscle. Usually, the skeletal muscle is attached to bones by tendons. They originate they're closest to the trunk and or more stationary bones. Insertions are more distal and mobile attachments. Flexors bring bones together. Extensors move bones away, and antagonistic muscle groups are like the flexor-extensor pairs. So just to recap again, flexion and extension. Flexion is when you move the bone closer together. So for like our example, when you're doing a like a bicep curl, that's when you're flexing the bicep, and at the same time the tricep relaxes. And then when you are extending your arm at the end of that bicep curl, the bicep muscle relaxes, but the tricep muscle contracts. So now let's talk about the anatomy of skeletal muscle. So these muscles are called muscle fibers. They are fused with many nuclei. Their satellite cells are also called stem cells, and they differentiate into muscles when needed for growth and repair. Cells are also bundled into fascicles surrounded by connective tissue. And the skeletal muscle fibers are the largest cells in the body. So fiber internal structures inside of the skeletal muscle would be the sarcolemma, that's like the cell membrane. You have the sarcoplasm, which is like the cytoplasm, sarcoplasmic reticulum, which is similar to the endoplasmic reticulum, but it wraps around each myofibril like a piece of lace. And you also have a network of transverse tubules, also known as T-tubules, and they're connected to the sarcolemma. The myofibrils inside of the fibers contain contractile structures known as actin, myosin, and accessory proteins. The actin are the thin filaments, myosin are the thick filaments, and the accessory proteins are troponin and tropomyosin. So this is just a diagram, detailing like what I just read to you before. So the transverse tubules, they're right here, they're also known as T-tubules. They bring the action potentials into the interior of the muscle fiber. And the you can see sarcolemma is this like line right here that outlines it, acting similar to the cell membrane. And then you have the sarcoplasmic reticulum, which is this, and on the inside has little connector tunnels that look like lace. This area stores calcium, and on each end is a terminal cisternae. On the inside are the thick filaments, which is this deep purple line that's known as myosin. And then you have thin filaments, which is this red, kind of like thread-like interwoven lace or interwoven area. That's the actin. And when you have two terminal cisternae, or two ends of the sarcoplasmic reticulum, along with a transverse tubule, that's called a triad. So structure of the skeletal muscle, you have the tendon and the overall skeletal muscle. And inside of each skeletal muscle, you have these muscle fascicles, which are a bundle of fibers. And inside the bundle of fibers, you have individual muscle fibers that have the nucleuses or nuclei on them. Ultrastructure of individual skeletal muscle fibers. This is just a closer look at the muscle fibers. So on the outside, you have your sarcolemma, and you also you have your transverse tubules that lead into the sarcoplasmic reticulum. And inside of the sarcoplasmic reticulum, you have these bundles or myofibrils. And inside the myofibrils, you have thick filaments known as myosin and thin filaments known as actin. So looking at a thick filament more closely, you have these myosin fibers or myosin molecules right here. When we magnetize it or look at it in a magnified view, you have the myosin molecule. All of this is a myosin molecule. And you have the myosin tail. This is the hinge region, and these are the myosin heads. For the thin filaments, or also known as the actin chains, you have tropomyosin, which looks like a thread, like a two-piece thread. You also have troponin, which are these brown nodules right here, or receptors. You also have G-actin molecules and nebulin that runs on the inside. And something else that's specific to thin filaments is titin. So titin is a huge elastic protein that extends from the Z disc right here, and it looks like a Z to the M line. So the titin runs from the Z disc to the M line right there in the center. It stabilizes the position of the contractile filaments. It also helps return the stretch muscle to a resting length. And then the nebulin, which is inside of the actin, that's an inelastic protein that aligns the actin. And this is a diagram I found so that it can help you organize the information that I just covered, so you don't agonize over the information. So feel free to draw this out and revisit this on your own time. All right, so looking at a sarcomere a little bit more closely, one sarcomere is between two Z discs. You have an I band, an A band, an H zone, and an M line. So the I band is made of thin filaments or just the actin. The A band is made of the darker regions where the actin and myosin overlap each other. The H zone is a clear band in the middle where the myosin filaments are, and only those filaments. And then you have an M line that represents the proteins to which myosin filaments attach to. This is a diagram that shows everything that I talked about. So the sarcomere is really from this I band to I band. And inside of an I band, you have the Z line right here, and you have the actin and myosin where they stop, and then the actin and myosin where they begin inside of the sarcomere. So you have your I band, which is end to end and it includes a Z line. Then you have an A band that starts where the I band stops. So the A band includes the actin and myosin and where they overlap, as well as this M line. So inside of the A band is the H zone, and that would be this clear, sort of like a clear region, but it's only where myosin filaments are. And then the M line is right there in the middle. And this is just another diagram showing you the organization of the sarcomere. And then this is a diagram showing what a sarcomere looks like during contraction. So at the top right here, this is when the muscle is relaxed and everything is just kind of stretched out and in its relaxed state. And then at the bottom, this is when the muscle contracts. And so the H zone and I band both shorten. So this I band right here shortens, and the H zone, this yellow portion, shortens, while the A band remains constant. So the A band does not move. This purple highlighted region does not move, but the H zone, the yellow highlighted portion, does contract, as well as the I band, this red portion, does contract. All right, moving on to the excitation contraction coupling section of this lesson. That refers to a combination of electrical and mechanical events in skeletal muscle and is comprised of 4 major events. The first one, acetylcholine, is released from the somatic motor neuron. In the second step, the acetylcholine binds to receptors on the sarcolemma, and during this step, sodium influx exceeds potassium efflux, causing depolarization of the muscle fiber membrane and creating an end plate potential. The third step, you have action potential triggers calcium release from the sarcoplasmic reticulum. And this step, it's mediated by two membrane proteins, a voltage sensing L-type calcium channel dihydropyridine receptor and a ryanodine receptor. And in the final step, calcium binds to troponin and initiates contraction. So let's go through this excitation contraction coupling more a little bit further. So step one, or in this step right here, in the first one, somatic motor neuron releases acetylcholine at the neuromuscular junction. This net entry of sodium, because that opens up these channels, the net entry of sodium through acetylcholine receptor channel initiates a muscle action potential, similar to like a wave of depolarization. The action potential in the T-tubule or the transverse tubule alters conformation of the DHP receptor, and the DHP receptor opens the ryanodine receptor in the sarcoplasmic reticulum, and calcium then comes into the cytoplasm. The calcium binds to troponin and allows actin and myosin binding, and the myosin head executes power stroke. After that, the actin filament slides towards the center of the sarcomere. So this dotted line, it moves from this dotted line right here and over into the center. All right, and then after the contraction is completed, then the sarcoplasmic calcium is released back into the sarcoplasmic reticulum. This decrease in free cytoplasmic calcium causes calcium to unbind from troponin, and then the tropomyosin recovers, and the tropomyosin is this thread-like line right here. The tropomyosin recovers the binding site. When myosin heads release, the elastic elements pull filaments back towards the relaxed position. So sorry, towards the relaxed position. So from the red region back to the dotted region. And moving on to the sliding filament theory. Remember I mentioned the power stroke, so I'll just talk about this a little bit further. There are three major events in the sliding filament theory. You have calcium that binds to troponin, ATP hydrolysis by myosin ATPase, and actin and myosin sliding past each other during contraction. So right over here, the calcium levels increase in the cytosol. The calcium binds to troponin. Troponin calcium complex pulls tropomyosin away from actin's myosin binding site. The myosin binds strongly to the actin and completes the power stroke, and then actin filament moves. So just look at the sliding filament theory and an actual cycle. You have the ATP that binds to myosin. The myosin releases actin. Myosin then hydrolyzes ATP via ATPase. Energy from ATP rotates the myosin head to the cock position, and myosin binds weakly to actin. And then you have the calcium signal, and that causes a power stroke, and that begins when tropomyosin, which is not shown, moves off of the binding site. And then the power stroke is initiated, and after that, the myosin releases ADP at the end of the power stroke, and ADP is released. And then you have a tight binding in the rigor stage right here, and it all repeats itself all the way back over. So contraction to relaxation, contraction to relaxation. All right. If you have any questions, please feel free to email me or reach out. I love hearing from you guys. And I do not want anyone to remain, confused about the information. So if you have any questions, please feel free to reach out. Thank you so much for joining me in this lesson. I will see you in the next one. Goodbye.